Hello, and welcome to everyone joining us for today's Select Science webinar titled Accelerating Bacterial Quantification, Exploring Rapid and Accurate Automated Cell Counting with the QUANTOM Tx. My name is Matilde Marques. I'll be moderating today's presentation, and I'm delighted to be joined by our guest speaker, Javier Garcia from Logos Biosystems. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of Auditorium and click to open the Q&A window. So, without further delay, I'm delighted to hand over to our speaker, Javier. I would like to thank him for presenting to us today.
Thank you very much for attending today to this webinar. We are going to see how we can accelerate the bacteria quantification. We are going to explore breakthrough the quantum Tx to ensure rapid and accurate automated counting. So my name is Javier Garcia. I'm field application scientist at Align Genetics. Logos Biosystem is part of Align Genetics. And we well know the problem and the challenges that we are facing when we want to count bacteria. We know how important bacterial counting is for any essential process in the microbiology labs, and how important it is to have accurate results because that's crucial for research or diagnostic. However, the different methods that are used in different kind of labs they are affected by the accuracy of the method itself because they are not so accurate. So in this presentation, we will explore the limits of those methods and how we provide a full solution to overcome those problems to count a bacteria automatically. In this case, with our system, we combine an algorithm and automated post-counting analysis, and this solution greatly increases the speed and facilitate the counting of bacteria with any loss of accuracy in your labs. So let's have a look to the content of the presentation today. As I said, Align Genetics is well known in the name of Logos Biosystem by the cell counters. The Luna family is our range of the cell counter, but we know the challenges that we have to face with the bacteria. So we are going to go through the classical method of the limitation that they have, and how rapid and accurate the automatic count can be using the quantum Tx, and the benefits that is going to bring you the quantum solution with the method that you're using in the lab. To prove that, we are going to have a look to different kind of cells, different kind of applications, and then we will see the benefits that we will get. Sometimes we need to think about how many bacteria there are there, because we know that the bacteria are the best, are challenging. So one of the first questions that we can address is how many names of species of bacteria are there. If we just check the literature, we can read that there are around 3,000 formally named species that are in pure culture and for which the physiology has been investigated. Those species now are being defined by PCR amplifying the ribosomal genes and then by sequencing. So the criteria to define different species is that the ribosomal gene is at least 3% different. But then if we think about what is known about the distribution of the bacteria species around the world, we can say almost nothing. We can answer almost nothing. We know sometimes about the biography of the infectious disease, as many of them are worldwide very well with a little population differentiation as the E. coli. But some of them are very specific to particular regions. But if we just want to be focused on how many species of bacteria are found in humans, I remember that we used to listen in the university that there are, in your body, we have around 90% of the cells are bacteria, and only 10% of your cells that you are walk around with are you. So, but they are so much smaller. So I don't really think that we really know how many species are found in humans. Just think about your mouth. Here we are talking about 400 to 500 different kind of in our mouth. So that's a lot. So that's give an idea of the complex sample that we need to handle when we want to quantify bacteria. Look to the bacteria under the microscopes. we know that they can be really diverse. It's a diverse group of microorganisms, and the diversity arises due to various factors such as genome variability, horizontal genes transfer, mutation, adaptation to different environment conditions that they allows them to colonize diverse habitats and different niches. So there are different methods to count bacteria, but due to the high variability, some of them just provide a close estimation of the real number of the cells that we have present in the sample. Some of the limitations that we're going to have for the classical methods must to be addressed to find faster solutions to quantify bacteria more accurate and reproducible. So let's have a look to the different classical methods. The classical methods and limitations. So we are going to see the limitation of the classical methods, but first we can classify as dilution methods. This is quite important because those classical methods doesn't comply the requirements that any lab would like to have. Because the dilution method, what we are going to do is to make serial dilutions, because it's a technique that for bacterial counting that involves diluting the bacteria sample in a series of steps to obtain a certain concentration that can be easily counted. This method is widely used in microbiology and is relatively easy to perform. To perform the series dilution, a small volume of the bacterial sample is going to be mixed in a known volume of sterile diluent, and then the mixture is going to then serial dilute it by transferring it to the small volume of the diluted sample to the next tube of the diluent. So that makes possible to create different kind of dilutions. The final dilutions is going to be placed onto the solid growth. So we are going to spread here in the different petri dishes, and with the medium that is necessary for those cells, we wait them for days to grow. So then the colony grows, and they are going to be counted manually to determine the concentration that we originally have in the different dilutions. So this is a method commonly used in the labs, but however, there are different kind of limitations. One of the limitations is the labor intensive. We know how difficult it is or how expensive it can be using different kind of medium for different kind of bacteria. But the most important point here is the time consuming. It can take days to wait until the cells grow as making colonies. But also the large volume of glassware that we're going to use in terms of plastic, we're using a lot of plastics. But there are also many factors to control: temperature, humidity, and contaminants. This is really relatively easy to contaminate those petri dishes with another bacteria. So at the end, it doesn't give the real number. It's just an estimation of the cells that you have there that are making colonies. So let's have a look to the next classification. For the indirect method, we can think about turbidity, metabolic activity, right weight, and current flow. So the turbidity is using a light source and a spectrophotometer where to quantify the light that is reaching the detector of the spectrophotometer when it's passing through an inoculate broad culture tube. So that means that we are just measuring the concentration of the cells that we have in the tube. By the metabolic activity, we can measure the amount of certain metabolic product that is correlated in direct proportion with the number of bacteria. The dry weight, what it's going to do is to just filter. The first step is going to be to filter and using a scale, and this method is widely used for measuring the growth of the molds. Imagine that we need to have certain quantity of bacteria here to quantify the number of bacteria that we have. But also, if we have some kind of other material there present in the sample, the weight is going to be also measured that contamination. Another method can be the current flow. This method requires a special system to measure the current and calculate the concentration of the bacteria that is present in the sample. So the limitation that we can talk about here is that must to be combined if we want to have an accurate data with colony counting, knowing the time and the money that it will take, but only count total cells, and it requires a high concentration of bacteria. So that means that knowing all those limitations based on the different kind of methods, those methods provide low accuracy if the sample is contaminated. The next method can be the classical methods. For the classical method can be the direct methods. So in this time, we are going to perform direct counting in a small volume of the bacteria sample that is placed into the microscope on the slice. Then the cells are counted under the microscope and is calculated based on the volume that we place into here to know the final concentration, because manually we are going to count just visualizing the cells under the microscope. Using an emulsimeter is another widely used method to count bacteria in the lab. However, it's also that has limitations that can be affect the accuracy of the count. Having a look to the limitations, the result depends on each analyst. So it really depends how good you are counting the cells. And we know that sometimes they combine two different people just count to see how many cells they are counting, and then they take the average. So that means that you know that you are using a lower accuracy in your method, and that means that it's labor intensive and time consuming. And again, we are here only counting the total cells. But there are labs that are lucky, they have more budgets, and they can have a flow cytometer. If you have a flow cytometer in the lab to quantify bacteria, this is a newer method to count bacteria because it involves passing that bacterial cells through a laser beam in the instrument. So the laser beam is detecting the scattered light to determine the cell count. But again, we have here some limitations. The limitation is based on the size of the cells that can be counted, not because any kind of cells, any size of cell can be counted, but also when we have complex bacterial population, can be tricky to handle. But here the more tricky point is when those cells are making clusters. If the cells are making clusters, those clusters are going to be counting as individual events, meaning that it's not going to provide accurate results. But also it has some limitation to viable cells if you want to measure viable cells, because the system is just counting any cell that is passing through the laser beam. So we cannot have the information on how many viable cells we want to measure. It's tricky to measure this. But it's also something to consider that this method is expensive. Not any lab can have it. So based on those limitations, it's well known that we have limits to address and to find alternative solutions to speed up the process of quantifying bacteria. This is well known. So having a look to the literature, we can find on the European Pharmaceutical Review, especially in the United States Pharmacopoeia as well, that the general information in the chapter 1, 2, 2, 3 describe the validation of alternative microbiological method that became official in December from 2015. This section contains the discussion of the limitation of the method that are validated as the colony forming unit. Because we know that there are many other signals that we can get from the cells that can be measured to quantify more accurate those cells. So this section highlight that is inappropriate and is a gold standard method for validation where there are many signals available other than the CFI used to detect the cells. The enumeration and the identification of those microorganisms in water or air or any other samples should be more accurate with new technologies. So the question is, what is the impact that is going to have when you're using those methods that is costing you a lot of time, money, and it's not accurate? For instance, when you are working in a pharma company, and the result, it really gives you the accurate number of the bacteria that you have. So we can conclude the current methods for counting bacteria in the labs, they have limitations that can affect the accuracy of the counts, but there are solutions that help scientists, researchers, and anyone working with bacteria to quantify them. As the technology is still continuing to grow as the bacteria grows, Logos has developed a faster and efficient and more accurate method to count bacteria in any lab, leading to better research and diagnosing in microbiology in a faster way and more accurate. So today we would like to present the QUANTOM Tx. The QUANTOM Tx microbial cell counter is a full solution for rapid and accurate counting of bacteria. At Logos Biosystem, we know the challenges that must be addressed, and this is why we talk about solution. It's a full solution that provides accurate and rapid counting. Having a look to the challenges, we can see that there are a few challenges. We can classify, if we want to make, let's say, a easier classification of the different kind of cells, we can see that some of them they have different kind of genomes. They have different genome size, but also they show different cell walls and membranes. So what we do is we provide two different kind of kits, the total cell staining kit and the viable cell staining kit, to stain the DNA that will provide a fluorescent signal, and those dyes must to pass through the different membranes of the different cells. So this is the solution that we propose to stain the bacteria that allows us to quantify the total cell number in cells per ml, or using the other kits, we can have the viable cells. And also the system will give you the information in cells per ml. But when those cells are stained, they are still moving. There is some kind of mobility of the cells. So to approach this problem, to approach this challenge of the cells when they are moving and make possible to be counted, we use a specific buffer, and we use a step of centrifugation to put all the cells in the same focal plane and to detain the cells and ensure that we are going to be on focus when we count the different field of views. But there are also another challenges, the cell size and the morphology. We have seen that the cells can be in different sizes, but also they will show different kind of morphologies. So based on the images acquired with the high sensitive camera that we have in the QUANTOM Tx, we will acquire images, and the algorithm and the image analysis process will detect any fluorescent signal that is coming from the cells, because those specific dyes will only stain our bacteria. So the algorithm is able to distinguish, is able to tag any single cell that is present in your sample. But even if those cells are making clusters, as we can see here, when we have clumping cells, we have seen that the flow cytometer will count this as a single event. But here, when we have clumping cells, the advanced algorithm with different declustering levels make possible to distinguish each cell. The protocols that we use and the two kits that we have to count the cells is the total cell staining kit and the viable cells. So let's have a look to the quantum total cell staining kit, how it works. So the first steps involve to take your cells, so you take the cells where you have it, and you would take 10 microliters of your sample that contain your bacteria, plus 1 microliter of the dye, plus 1 microliter of the enhancer to increase the fluorescent signal. Sometimes for the total cell staining kit, we don't need to incubate it. It's not the most common case, but let's say that we take it for a few minutes, and then we can add the loading buffer. The loading buffer, we are going to add 8 microliters in the ependor tube. We will mix it, and then slowly and carefully, we are going to load with the pipette 6 microliters into the slide in each chamber. We will have 2 chambers per slide. This slide is going to be centrifuged in step 2 to immobilize the cells and to put them all of them in the same focal plane. This step of immobilization with the centrifugation step, it will take 10 minutes. After that, we just take the slide and we insert it manually into the QUANTOM Tx, the automatic system to count the cells. This system will take 10 field of views, so it means that we'll take 10 images to detect the cells that are present in those 10 field of views. Considering the volume that is measured in those 10 field of views, and considering the dilution factor based on the dilution that we apply here, at the end we will have a PDF report with all the information. We will know the final concentration, in this case for the total cells expressed in cells per ml. Also, we will have all the details of the protocol used here in the QUANTOM Tx to count the cells. But also we will have the raw images and the tag images to see the cells that has been detected by the algorithm. So we can really see if the quality of the counts is well done or not. But also we have another kit. We have the total viable kit. When we use the total viable kit, means that we are using incubation step. For this incubation step here, we can say that it will take, before with the total cell staining kit, it will take less than 30 minutes. But with the incubation step here for the viable cells, it will stain only the viable cells. So we are only counting cells that are alive, wherever they make colonies or not. Any cell that is alive, we are going to count it. That's huge. That's a huge step. That's quite important. So the time that will take for us in the total timing for counting the cells is going to be around 42 minutes. Let's say less than 40 minutes. Sometimes it requires, some cells can require longer incubation time, but no longer than 1 hour. So we can see that for the total cell staining kit or the viable cell staining kit, we are talking about less than 40 minutes to count the cells. That brings a lot of benefits that we can resume together if you're working in different kind of labs. When you work in different kind of labs, those are the benefits. If wherever you are working in public research, startups, hospital research, pharmaceutical industry, when you have a nice idea for a startup that goes for the food industry, or even if you're working in health safety agencies. So the Quantum Tx is going to bring benefits for those labs. If we compare the classical method with the Quantum Tx and the benefit that it's going to bring, we can just go again through the classification that I used before to see how different methods can be improved or the benefit that it's going to bring if you use the Quantum Tx. The first classification was the indirect methods. So in the indirect methods was the turbidity, metabolic activity, dry weight, and current flow. So having a look again to the limitations that we have here with those methods is that must be combined with colony counting to increase the accuracy, only count total cells, and it requires high concentration of bacteria. But also we know that it has low accuracy if the sample is contaminated. So the benefits that we are going to have is if we use the Quantum Tx is that provide the total cells and the viable cells concentration if we use different kits. But also a wide range of bacteria concentration can be measured into the Quantum Tx, providing a high accuracy and reproducible result in a really short time, meaning that we are really fast. If we have a look to the direct methods, then we can think about again about the emulsitrometer plus the microscope. As we know, the result depends on each analyst, it's labor intensive, time consuming, it only counts total cells, and it has again low accuracy. With the Quantum Tx, the benefits that we are going to get if we replace those methods by our system is an easy sample prep, automatically counting for the total and viable cells with a high accuracy and fast and reproducible. If we have a look to the dilution method for the serial dilution plating or spread plate method when we want to have the CFUs, this isn't the most common method that must be improved. It's widely used because it's simple, it gives a good, let's say, general idea of the cell viability, but it's sensitive even for a low concentration of bacteria. But however, this method is dependent of the grow media and the condition as well of the being limited by the cultural bacteria. That means that which exclude viable cells but no culturable cells, strains. So meaning that if the cells they don't have the nutrients or the media that they need to grow, even if they are alive, we are not gonna detect it because they are not making colonies. Another major disadvantage is that it takes days to get the results. It takes really a lot of days to get the results. One colony may arise from one, so some colonies takes a lot of time. It's crazy the time that can take. So the time that you invest is significant drawback when you assess these time-sensitive issues when you are working in the food water industry or you are working with diagnosis that you need to have the results as fast as possible. So the quantum is going to bring a lot of benefits. It's going to be so much faster. We are talking about minutes instead of days. It has an easy sample prep. We are not going to use a huge volume of plastics in the lab, and it provides the real concentration. It will give you the total cell number per ml that you have there. It's accurate and reproducible. If we have a flow cytometer, when we have a flow cytometer, the benefit that is going to bring by the Quantum Tx is that the range of the set that can be counted is a broader range because the set that can be counted is broader. But also, we have seen one of the limitations of the flow cytometer is when the cells are making clusters, it doesn't decluster the cells. The Quantum Tx is able to decluster those cells and detect any single cell that are making clusters. So that means that it's going to provide you more accurate results wherever you use the total cell counter, the total cell kits, or the viable cell kits. It's less expensive and it's so much easier to use than any flow cytometer. We have been talking about a lot accuracy, so how we can prove that we are accurate? The Quantum Tx brings benefit for the labs, and one of them is the accuracy that you can get when you have the total cell count or the viable cell count. So counting accuracy of the quantity was confirmed here using different kind of dilutions. So that means that we will have different concentration factors. And at the end, those measurements with the total cell concentration that we get in the Quantum Tx, it provide a strong correlation between those. So it means that we have here a high accuracy and a strong correlation of the theoretical data that we should get. So this is a proof how accurate we can be using the Quantum Tx. But also it's important to know the numbers that we get using the Quantum Tx, how they looks like compared with another methods. Another method used in the labs that we have seen can be the flow cytometer or the MO cytometer. The flow cytometer is recognized as one of the most accurate ways to determine the total and viable cells count. But we have seen that is critical if we have cells that are making clusters. So to use a flow cytometer, it requires to have an expensive instrument, but it's required to have a training operated person that is able to count those cells. So if we compare this data that we get on the left when we make different E. coli suspensions and different dilutions, we can see that the Quantum Tx shows comparable results and accuracy as the flow cytometer if we are using single cells that are not making clusters. But if we have a look to the MO cytometer, it shows that there is high variability when we count it. So let's have a look to some kind of applications that can be used, that can be run on the Quantum Tx and see the benefits that we can get. The Quantum Tx can work. In this case, we are working with a total cell staining kit to provide the final cell concentration of what we have in the sample, even if we have contaminated samples. Some of the applications that we can think about is when we would try to develop bacteria strains with some kind of capabilities of digesting different kind of material that can be wood, plastic, or any other ones. Therefore, there's a high interest to develop solution that will speed up the process of the degradation of the material that is contaminating the environment, the different mediums. So here, if we have a look to the image provided by the Quantum, we can see how specific are our dyes to stain bacteria. In this case, we are using the total cell staining kits. And we can see how the system is tagging the cells that shows fluorescent. But when those cells are in presence of cellulose fiber, this is not detected as a bacteria. So this provides a more accurate counting even when we have contaminated sample or if we have debris. So those kind of customers, they used to use CPU with the time consuming that we know that it will take, but also they used to use turbidimetry with the low accuracy that is provided. So in terms of speed and how fast we can get the results, we are getting the result in 30 minutes compared with the 48 or 72 hours that will take when we use the CPU method. But even being faster, we can be more accurate and reproducible. A look to another application can be the study of the intestinal ecosystem. When we think about that, now we are thinking about microbiota and probiotics. So studying microbiota and probiotics is important for several reasons. We want to maintain a healthy gut, so the microbiota plays a crucial role in maintaining a healthy gut and overall health. So by studying those microbiota and probiotics, scientists can better understand how important it is to maintain in good balance those microorganisms in the gut to have a good health. But also it's important to prevent and treat the disease that when we have imbalance of those microorganisms. But also if we want to develop new therapies, studying microbiota and probiotics, scientists can better understand how to develop new probiotics therapies to treat a range of health conditions when they are not in balance. So that means that we are talking here about balance of the different microorganisms, so we can think about how important it is to have an accurate number of cells that are present in the sample. Another application can be improving the food production. So those microorganisms are involved in many food production processes, such as fermenting dairy products as cheese, milk, different kind of brewing beers, making bread. So studying those microbiota helps to understand how we can make it faster, and how we can have those conditions in a safer process to improve food safety, quality, and speed of the results. So those customers usually are using CPU and turbidimetry. Again, it's taking long and it can be really low, it can be really slow. So the benefit that we can get is that we can get the result in 30 minutes compared to 24 or 72 hours. The report that we can have, those images become from the quantum, has been provided by the quantum from Propioni, Bacillus, but even if we have facing samples, with this complex matrix, we are able to count the cells because the dye are going to be targeted to the cells and it's going to provide a fluorescent signal. It's going to be detected by the sensitive camera of the cell counter. So that means that we have high accuracy and reproducible results with an automatic method provided by the solution of the QUANTOM Tx. But even like that, if we have a specific sample, look to the lactobacillus. When we have a look to the lactobacillus here, what we try to prove is to have an expected concentration in CPU per ml, and also the total cell concentration in cells per ml. So here in the first line, we have the raw images that is provided by the quantum, and in the second line is the tag image provided as well by the quantum when it's detecting the cells, when it's detecting the fluorescent signal. So having both images allow us to, let's say, to qualify the detection of the cells done by the system, to check if the system is missing cells or not. But also it will give us information in the future to track, to have a traceability, how the count has been made in the past or how it should be done in the future. So the correlation that we can have here between the cell concentration in cells per ml and the CPU per gram, we can see that have a good correlation that we can see in this graph. But there are also another applications, maybe vaccine research. In vaccine research, we can think about lestosphere. So lestosphere usually is quantified in different labs using an hemocytometer and the microscope. Or in the best cases, you can have a flow cytometer. Here, if we use the QUANTOM Tx to quantify lestosphere, we can see that after the centrifugation step, if we zoom into this image, we can really see if the cells are horizontal or vertical. Even like that, we are able to detect it. So the way to get the results here is faster. We provide an automated method, and we can standardize the counting method for lestosphere for vaccine research, where the speed, where to having fast result is quite important. But also for those people, it's important to have the PDF report for monitoring the counts and to have the proof of the different count that has been made. Because when we use an hemocytometer, you don't have any proof how anyone was counting the cells. Another nice application, and maybe it's quite important because the medium that we are using is quite expensive, are the mycobacteria. We know that the mycobacteria need to have an expensive medium to make them grow. But why is so important to study mycobacteria? There are important reasons. We can think about the public health, because some species of mycobacteria, such as the Mycobacteria tuberculosis, are responsible for causing serious disease in humans. The tuberculosis is a major global health problem and is one of the top 10 causes of death worldwide. So understanding the biology of the mycobacteria is essential to develop effective treatments and vaccines to combat this disease in a faster way. But also it has an important role in the environment because the bacteria are also found in the environment that makes an important role because is breaking down the complex organic compounds that is going to contribute to recycle nutrients in the ecosystem. So this is why it's so important. But also in biotechnology, because in biotechnology, mycobacteria has unique properties that make them useful for biotechnology applications. For example, the Mycobacteria segmentatis is a commonly used model organism for genetic studies. And also the Mycobacterium bovis that is used as a vaccine against tuberculosis. So we can see how important they are. All of us, we know how important it is to develop new antibiotics because the bacteria can change easily and really fast. So they can express and resistant to the medicines that when we try to treat any kind of disease. So in summary, it's so important, this topic, the mycobacteria, to speed up the process to quantify and to have accurate counting when we use the quantum Tx. So let's have a look to one example. In this case, we can have customers that they are using different concentration of the bacteria, and they just take one example. They take one sample from here, and they measure how much they have just by turbidimetry, and then you can just prepare the inoculum that is going to be injected into the mice. Those mouses will have different concentrations. Some cases we can have the wrong concentration or the correct concentration. But at the same way, in parallel, we are going to take the control. We are going to take exactly the same concentration that was here to grow the cells here and to count the colonies. So after that, after 4 weeks here plus 4 weeks here, we can see how was the treatment of the different mice to see how was using different kind of controls, if the treatment was satisfying our needs or not. So our goal. So that means that these validation studies using animals, it has a high cost, and it takes a lot of time. We are talking about 4, 6, 8 weeks. And also here we're using animals. For this, we can also use the quantum Tx. If we use the quantum Tx, it's going to bring a lot of benefits. The benefit is that instead of using weeks, we have seen, let's say 4 weeks for the best cases, but can go up to 8 weeks and using animals. Instead of that, without using animals or without using many plastics, it will take us only 15 to 45 minutes. It depends if you use the total kits or the Bible kits. It's an automatic method. So imagine just few minutes compared with weeks. And also it is safer, accurate, and reproducible. The problems when you use the CPU is that it's not accurate because many of those cells are dead and they are not growing, they are not making colonies. But this problem can be easily solved so much faster with the use of the quantum Tx. If we have a look to the images, those are the images how they look like under the quantum Tx. So the high sensitive camera using our specific dyes and the protocol that we propose to prepare those kind of samples to face and to find solution of the challenges that we already explained shows images even at low concentration where we cannot almost cannot see here anything, but here we can see a small set that is detected by the quantum. Even at low concentration or high concentration, different kind of species can be detected using the different kits with the Quantum Tx. So here we propose a nice solution that will help any scientists that are working with bacteria, in this case with mycobacteria, to quantify the cells. As a result, we have a full solution for a rapid and accurate counting of the bacteria, because the result can be get in minutes compared with days or weeks that you are usually having, but it's an automatic system. It doesn't depend on the technician that is counting the cells, but it will also provide the total cells and the viable cells depending on the kit they're going to use. The Quantum Tx is using a high sensitive camera to detect the signal that is coming from the dyes when those dyes are linked into the cells. Here we use low volume of glassware. We just use few tips, the slice, and the tubes to prepare the samples. And the result that we are going to get is going to be accurate and reproducible. This is a touchscreen system that is with an easy user interface that makes possible to count 10 field of views of the cells. So that means that we increase the volume of counting to have more accurate results. But also this system can be connected by Wi-Fi to your PC to have a look to the data. So you will have access to all the information that is saved into the Quantum Tx from your PC. You enjoyed the webinar, and this will bring you new ideas and new solutions to speed up the process and to help you to have more accurate and reproducible results when you are counting bacteria. Here from Align Genetics, from Logos Biosystem, we are glad to support you and to provide you more information in the future if it's needed. Thank you very much for your time.
Thank you, Javier, for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We'll now move on to the last part of today's webinar, the Q&A session, which I'll be handing over to my colleague. As a reminder, to ask a question, you just need to simply head to the engagement window to the right of Auditorium and click to open the Q&A window. Thank you.
And so the first question we have for today is, how does the Quantum Tx respond to Staphylococcus aureus? Are there strains of staph that have reduced clumping? And have you assessed these on the Quantum Tx to assess differences in clumping versus single cells?
Okay, thank you for the question. This is quite interesting because actually we have two articles where we can see that the scientists, they have used the Quantum Tx to quantify the Staphylococcus aureus, so it's working fine. So I can answer yes.
That's great, thank you. So we'll move straight on to our next question, which is, someone is wondering if you could elaborate a little more about the staining steps. And will the staining distinguish live and dead bacteria?
Okay, just to make a resume how the kits works, we said we have two different kits. We have the total cell staining dye, that is a fluorescent DNA binding dye. So you're going to stain to the DNA, and it will provide a fluorescent signal that will be detected by our sensor camera using the filters. But for the live cells, we are also using a permeate cells converter. So the dye is going to permeate into the live cells, and this will be, by hydrolysis, we provide a fluorescent signal that we can quantify to detect the live cells.
That's great, thank you. And it's great to see lots of great questions coming in. So we'll move straight on to our next question, which is the Quantum Tx system also compatible with other bacterial live/dead dyes?
Yes, we will just use some kind of relevant samples during the presentation, but you can see on our website, on different articles that you can find on field, there are many types of bacteria that people have using successfully to quantify bacteria. So if you visit our website to align-genetic.com, you can really see the list of bacteria, some of the list of the bacteria that have been tested.
Excellent, thank you. And someone has asked, are you working on a plate format, for example, 96 well?
No, not really. What we do is we want to quantify the bacteria. It was explained during the presentation. We use a specific slice where you need to load the cells with the specific kits. And then after centrifugation, you will use those slides to quantify the number of cells in the volume that we know that is there in the field of view that we are measuring. Actually, we are taking 10 field of views to measure the concentration of the cells that are there, and then using the factors based on the dilution that you made, then you will have the final concentration in cell per ml.
Great, thank you. And just a reminder to our audience that if you do have any further questions, please do send those in. You can submit them by simply typing into the questions icon to the left of your screen. And so the next question we have is it possible to do viability measurements with the Quantum Tx?
And not really viability, but if you want to do that, what you have to do is to measure 2 times. You have to use our 2 kits. First, you need to quantify the total cells. You need to use the total cell staining kit to quantify the total number of cells. And then you need to prepare a second sample to quantify the viable cells. And then you can make your maths, you can make your numbers, and really see the viability of the cell that you have there.
Excellent, that's a great answer, thank you. And what size cells can the Quantum Tx count?
Okay, actually the camera that we have, the sensor, is ready to quantify cells between 0.3 and 50 microns. So those kind of cells of those size can be counted without any problem.
That's great, thanks. And what is the cell concentration range of the Quantum Tx?
Okay, thank you for that, because this is quite interesting. This is why on the protocol we use the dilution factor, because we know the linear range where the system is working perfectly. But we can say as a resume that the Quantum Tx can sample with concentration range of 200,000 of cells per ml to 1 billion of cells per ml. But if we talk about let's say the optimal range, I will say something around 1 million of cells to 500 million of cells per ml. But as I said, using the dilution factor, the system will finally give you the right number of the cells that you have there. So you don't need to make any extra calculations to know the final concentration that you have.
That's great. And just a reminder to the audience that you can also download related resources in the tab to the left of your screen, and this includes the certificate of attendance. And so we'll move straight on to our next question, and that is, what affects the staining time?
Okay, usually, as this was explained during the presentation, when we use the total cell staining kit, we don't need to have, let's say, an extra incubation time. But for the viable kits, we need to optimize the staining time. So because some cells are more resistant for the penetration of the dyes, so in those cases we need to work in higher incubation time. So how is the effect? What is the effect that we are going to have? The effect is that we need to know we need to have the optimal timing to have the optimal staining of our dyes. If you increase this time, nothing happens, but at least you want to optimize the workflow. So as shorter is the incubation time, faster you will be with your counts. We know that we have a shortened counting time compared with another techniques, but let's say that something as a maximum of half an hour for the live cell staining kit is more than enough to stain the bacteria you want to quantify with the Quantum Tx.
That's perfect. Thank you for that answer. And just a reminder that if you do have any further questions, please do send those in. You can submit these by clicking into the questions icon and typing in your question. Great. Well, I think that's actually all we have for today. We have one more question actually, sorry, that's just come in. So this question is, can the system use 96 well plates? Is the output template / file format customizable? And can we use the Quantum Tx to count CFUs on a membrane filter plate?
Okay, as I said before, we develop specific slides, so you can load up to two samples in one slide to be counting on the Quantum Tx. So the system is not compatible with the 96 well plates, but you can prepare in the centrifuge up to eight samples at the same time. So 8 slides, so that means 16 samples. And the output files that we are going to provide, when you make the counts, the system will save directly in the SSD drive that is internally in the Quantum Tx. It will save the images, the raw images that is taken by the system. So that means 10 images per sample in raw image with TIFF format. The tag file just tagging the cells that has been counting, where the fluorescence has been detected. and also the PTO report with all the information. Additionally, we also have the possibility to extract information CSV file that will help also to have the resume of all the counts that you have made on the Quantum Tx. Thank you.
Excellent. Thank you very much, Javier, for that answer.
Well, I'm afraid that is all we have time for today. I would like to thank Javier for today's discussion and presentation and this lovely Q&A session. And thank you to everyone joining us online for submitting your questions. We hope that you have found this a worthwhile session. If we didn't quite get to your question today, we'll follow up with answers to these after the webinar. And if you have any other question after today's session, please feel free to email myself at editor@selectscience.net. If you are interested in receiving a certificate of attendance, this will be shared with everyone who has attended live today via email a few days after the event. And if you'd like to listen again to today's webinar or invite a friend to do so, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.